Hi friends, today we'll be talking about the de novo synthesis of pyrimidine nucleotides. The de novo synthesis of pyrimidine nucleotides requires aspartate, carbamyl phosphate and PRPP. The two common pyrimidine nucleotides, that is the CMP and UMP, have the pyrimidine rings of cytosine and uracil. The synthesis of pyrimidine nucleotides is different than that for purine nucleotide biosynthesis. The six-membered pyrimidine ring is first synthesized and attached to the ribose 5-phosphate. The process requires carbamyl phosphate. In animals, carbamyl phosphate for urea synthesis is synthesized in mitochondria by carbamyl phosphate synthetase 1. The carbamyl phosphate required for synthesis of pyrimidine nucleotide biosynthesis is synthesized in a reaction catalyzed by carbamyl phosphate synthetase 2. So there are two different enzymes for catalyzing the formation of same carbamyl phosphate in animals required for different purposes. Bacteria, they have a single enzyme which catalyzes the formation of this carbamyl phosphate for synthesis of both arginine as well as pyrimidines. The bacterial carbamyl phosphate synthetase is a big enzyme, a large enzyme which has 3 active sites, and it provides a best example of substrate channeling, wherein the substrate are passed from one active site to another without the loss of unstable intermediates. So let us have a closer look at the de novo synthesis of pyrimidine nucleotides.
In the first committed step for the pyrimidine biosynthesis, which is catalyzed by aspartate transcarbamylase, the carbamyl phosphate reacts with aspartate forming N-carbamyl aspartate. The bacterial aspartate transcarbamylase is the most thoroughly studied allosteric enzyme. In the next step of dehydration, the water is removed from N-carbamyl aspartate and the pyrimidine ring is closed by dihydroorotase to form L-dihydroorotate. The dihydroorotate dehydrogenase then catalyzes the oxidation to form orotate with NAD+ as the electron acceptor. The eukaryotes have these 3 enzymes, that is the carbamyl phosphate synthetase 2, aspartate transcarbamylase and dihydroorotase as a single trifunctional protein also called as CAD. The orotate phosphoribosylate transferase then attaches the ribose 5-phosphate to orotate from PRPP forming orotidylate. This then undergoes the removal of carbon dioxide, that is decarboxylation, by orotidylate decarboxylase forming uridylate. This then is phosphorylated by ATP in the presence of kinases to form UTP. For the synthesis of CTP, the cytidylate synthetase with the hydrolysis of ATP and the nitrogen from glutamine forms CTP. In many species, the cytidylate synthase can use NH4+ directly as the source of nitrogen. Thus both UTP and CTP are formed.